All right, good afternoon, everyone. I think we can get started. So thank you for joining us. I'm Chris Roche, Director of PIN, and I want to welcome you to this second workshop in our series here. And I'm happy to see a great turnout again. I appreciate everyone's interest, particularly as the semester has begun to ramp up. So today we'll focus on the impact of traumatic brain injury on the risk for dementia and Alzheimer's disease. And I'll give a quick introduction similar to some of the comments that I provided at the beginning of the last workshop, just to give an overview, particularly for those who weren't able to attend the first time. So the goals of the workshop are really to identify major challenges in the field of AD and other dementias that would best be tackled using an interdisciplinary research approach, much like we're often doing with these sorts of workshops in general. And then to create teams of Purdue faculty members who are really best equipped to solve these problems. The motivations are to build a community of AD investigators. We've actually begun to do quite nicely as far as increasing our expertise in this area on campus over the last couple of years. We'd like to continue in that direction and of course, add to the innovation in our research in this area and really the impact of problems that can be tackled by bringing together experts from diverse areas. Another point that I'd like you to keep in mind that's really on our mind, on the leadership team, is the idea that as we organize ourselves here at Purdue, we can then approach experts at the Stark Neurosciences Research Institute, which is really one of the best institutes in the area of AD research, really in the world, and present to them some compelling research programs where they could then collaborate with us and add to our capabilities, particularly in the area of preclinical models. Another point, of course, is related to funding. I mentioned this before previously that I think that with these efforts, we should be quite successful given the really attractive pay lines in the area of ADRD research at NIA and also at NINDS. So I think that the timing is right and we should capitalize on this attractive funding landscape. So for today's workshop, we're really continuing sort of, I think of this as a second part of the theme of environmental risk factors. We focused largely on toxicological risk factors in the previous workshop. And here we can think of traumatic brain injury as another environmental risk factor leading to an increased risk of Alzheimer's disease or other dementias. And I think this is particularly interesting from the perspective of opportunities for therapeutic intervention, because if you think about individuals who've sustained a traumatic brain injury, we know when their injury occurred, so we sort of know what the clock is in terms of events that could lead to neurodegeneration. And this is usually not the case except for...
There's only 6 people.
This is not usually the case except for individuals who might have rare genetic mutations. So I think that it really is something important for us to think about, particularly if we think about also the Purdue Institute for Drug Discovery, that there could be some translational opportunities here, particularly unique to the role of TBI in increasing the risk of neurodegenerative diseases, including Alzheimer's disease. So for today, we'll have 2 scientific presentations highlighting research in this area, and those will be recorded and posted, and again, posted on the PIN website with Purdue Career Account Access. And then I'm also very happy that today we have an additional presentation on federal engagement opportunities given by 2 representatives from Lewis Burke Associates, Ida Razzulli and Nicole Parker. And their presentation will also be recorded and posted on the PIN website. And then lastly, as we did last time, we'll have some breakout sessions, and the goal will really be for teams to coalesce through discussions in these breakout sessions. And here we'll post summary notes from these discussions. And actually much of the content from the previous workshop is posted now on the PIN website if you're interested and if you didn't have a chance to attend or if you'd like to review the previous discussions. Okay, let's see. Oh, here we go. So yeah, we didn't hear too much. I mean, I think the feeling was pretty positive with the last workshop, but I really do want to make the point that we're happy to make adjustments. This is still an experiment, so please feel free to send feedback anonymously, not anonymously, however you'd like, but we'd like to continue to improve through this workshop series over the course of the next few months. All right, lastly, to let you know about the current breakout session topics. So as we did last time, our two speakers have kindly agreed to lead two breakout sessions, preclinical models, which I understand will be both in vivo and cellular models in the area of TBI and AD. So Ree will lead a discussion in this area, and Sereno will lead a discussion on human studies. Chongli Wan has agreed to lead a discussion on the role of genetics and epigenetics, and Sebastian Hilly will have a breakout session in the area of computational neuroscience. If you want to navigate, so we've posted the web links in the chat box. You can also return to the main room at any time to have guidance if you'd like to get into different rooms, and feel free to either text me or Cynthia, and those numbers are also in the instructions that were emailed out yesterday. Okay, so I think that's all I have, and so then I think I'll stop sharing and I'll allow Rishi to start with the first talk. I think as we did last time, we'll have the two scientific talks without questions. The questions could be taken up in the breakout sessions, but for the talk given by the Lewisburg representatives, we will allow some time for questions. Reed, do you want to take it away?
Okay, can you hear me?
Yes.
Yes.
Okay, all right, thank you, Chris. Yeah, I think I'm gonna take a quick presentation to show some of the, actually mostly work in progress. So I think, so the idea is probably present this kind of a scenario that we can actually galvanize collaborations. So today I am going to focus on the pathogenesis of a TBI-induced Alzheimer's disease, and the focus will be really on protein aggregation mechanisms and beyond. So as we can see that the Alzheimer's disease is the topic of this, so I don't need to actually emphasize how important this disease is. Actually, it's the leading most common neurodegenerative disease. And in terms of the cause, I think we know quite a bit, but not obviously enough. So we know it's mainly caused by abnormal buildup of the protein, so-called protein aggregates, protein aggregation. Mainly the protein associated with Alzheimer's disease is beta amyloid and tau protein. So these two proteins. But today, of course, we focus on how this can actually, what kind of factors affect the Alzheimer's disease, and then in general, maybe how that affect, we focus on how that affect beta amyloid and tau protein aggregation. So age is one of the things, actually probably one of the most obvious important risk factor. But if you look at the risk factor I put out as a category as you can modulate, head injury is definitely one of the most important, and growing evidence show that this is actually very important to induce or trigger some kind of pathology which can ultimately lead to Alzheimer's disease. So far the evidence, the clinical evidence for Alzheimer's disease, both Alzheimer's disease and chronic traumatic encephalitis, CTE, is fairly strong, particularly I would say CTE, because CTE also, there's quite a bit of similarities with Alzheimer's disease, and also has a dementia as a component. So how we can do that, and what we the thought process is that we start from perhaps the primary and secondary injury. This is true for any CNS trauma. So we start with in this particular case, could be a blast injury, and then we see there's an immediate injury, but then there's a cascade of secondary injury. We believe that all of them perhaps has something to do with the protein aggregation, which can lead to the Alzheimer's disease-like pathology. So in a sense, we have a hypothesis here, so which is basically to study the mechanism of TBI-induced protein aggregation. We focus on aggregation. So perhaps we think maybe even physical impact itself could induce protein aggregation, but this could be worsened and also even the secondary injury can exacerbate the primary injury-induced protein aggregation, but also secondary injury, the chemical fact cascade can itself induce protein aggregation. So here's what we think, and then the third hypothesis we think is probably vasculature damage has quite an important contribution to this. Not only ischemia can cause inflammation, which itself can cause formation, perhaps aggregation of the beta amyloid, but also the damage of the blood-brain barrier perhaps can leak or deposit some of the circulating beta amyloid into the brain tissue, and then cause, of course, the accumulation of the beta amyloid in the brain tissue. So we started with a in vivo model. Again, we studied this in both in vitro and in vivo models. So this is two in vivo model that we have. One is the blast injury model. The other is in vivo concussive injury model. This would be probably the one on the right, maybe more related to the football player. The left is the soldier when they suffered the blast injury. So here we, so far the evidence that we have in terms of AD is that we discovered actually there's an increase of tau protein phosphorylation, which has been linked to perhaps the neuropathology, tau protein pathology, actually leading to Alzheimer's disease. This is actually true in particular in the CTE case. So there's a human evidence, actually clinical evidence to support that. Now, after that, we also think about that these protein aggregation probably can cause by these inflammation and also some perhaps even the chemicals such as aldehyde. We're going to talk about this a little bit later. But first of all, we also show that actually for this blast injury, we can see that chain and single and triple blast, there's a dose-dependent increase of inflammation. So if you look at the microglial cells, actually they are actually gradually can be more activated, and also this is in case this is again the blast injury, and then we're also looking to some of the chemicals perhaps can cause pathology. In this particular case, we measure the alkaline, which is the aldehyde, and this is only a single blast, and we divided the brain to about 53 different regions and discovered that quite a few areas increased, but not all of them, but quite a few. And one of them actually is hippocampus CA2 region. So this is area perhaps could be linked to Alzheimer's disease. And we know that this area, the alkaline level actually is significantly elevated. And then the other things in the in vivo model, we also like to know, because there's strong evidence to show that the vasculature damage, particularly long-term vascular damage, could be linked to dementia and Alzheimer's disease. So we take advantage of this particular model to, and this is actually the study collaborated with Yinji Tan, and he uses the near-infrared spectroscopy to detect lower level ischemia or lower level called hypoperfusion following traumatic brain injury and spinal cord injury. We already had some data to show that actually this is the case. As a matter of fact, with perhaps a single blast, we can see some damage, but they recover. But triple blast, we think that it will probably last longer. So this could be some kind of a biomarker we can detect, but also perhaps can study how this vascular damage could be related to beta amyloid formation. And we can look at actually the long-term non-invasive measurement. We also studied this with actually Tiffany Lau, looked at the BBB damage. In this particular case, we actually discovered that actually the parasites actually, as well as Desmond, there's some changes. In single blast, didn't see much, but in the triple blast, they actually, we saw changes. It's very significant changes, and then they recovered after a few days. So this is kind of interesting, and when we say blast, we only blast them for 3 times, but in reality, the repetitive injury could be much more numerous, could be maybe 20, 30 times. We can also study perhaps some of the comorbidities. For example, there may be something in a recovery period, if you don't actually rest enough, or there's enough time for you to rest, if you go back to play again, or be blasted again, this kind of recovery probably could delay or maybe never recover. So now, we also have a in vitro model to start this in conjunction with the in vivo study, is that we wanted to create a TBI on a chip to recapitulate clinical relevant phenomena. In this case, we culture the cell in a petri dish, and we can record the action potential, record the field brain activity at the same time, so we can subject this artificial brain to an impact. So in this case, we can look at the cells culture from, say, rodent, but also culture from human IP cells. So actually, these are the capability this particular model can actually offer. So in this case, we can already discover that upon impact, we see a brain activity. We call it brain activity. Actually, it's the brain cell culture in its artificial brain. And we already can discover they have an activity change. Also, in a sense, we also look at cellular damages. In this case, we saw a phosphorylated tau protein actually increase, phosphorylation increase, which indicate this a model could be studied, maybe perhaps the mechanisms, how this can be actually, what is the mechanism of actually this phosphorylation of tau. And we also look at the amyloid precursor protein to study perhaps what is the factors can influence this kind of a protein aggregation. In addition, actually, we also look at some of the chemical factors actually could influence this kind of phenomena. In this case, we also look at the aldehyde alkyl adduct. So discovered actually it's also increased. So indicate this kind of a TBI on the chip actually could be very useful, that you can study a lot of factors, actually how does influence the protein aggregation, but also study actually not only the chemical primary, but also the secondary injuries. And also this actually collaboration, in collaboration with Chris for a long time, but we also kind of duplicate or mimic this in this TBI on the chip. We give an impact, and we discover that alpha-synuclein aggregation elevation could be also observed in this particular model. Although the cell is a cortical neuron, so maybe perhaps in the future we can study something, maybe particularly we know it's a dopaminergic neuron, and perhaps we can even do some manipulations to genetically alter the cell and to study deeper into this mechanistic study, so understand how this alpha-synuclein aggregation happened. Yeah. I think I'm going to stop here, and I only present the data that actually, this is the collaborators actually who I present the data, and there's quite a few of them actually I did not present the data, but I will be happy to show them in the breakout sections. Yeah. Okay, thank you, Chris.
Excellent. Thank you very much, Ree. I think that gives a great overview of the preclinical models in your lab, which I know through all your collaborations are really available to anyone who's interested in this area. So thank you very much.
Sure.
Do you want to hand the reins over then to Anne next?
Yes. Yeah, and you can go ahead. Yeah, thank you.
Can you see?
Yes, yes. Maybe you can put a presentation mode, or if it. Yeah, that's good. It's good.
Perfect. Thank you.
Do you see the whole screen, or do you see it in? Okay.
Yeah, it's good.
Okay. So I'm Ann Serino. I'm a professor in psychology and biomedical engineering, and I'm going to talk a little bit about behavioral biomarkers. See if I can advance this. Alzheimer's disease, as Rihi mentioned, is a burdensome disease. It's burdensome in terms of disability as well as lengthy duration. It's burdensome for the individual as well as caregivers and society more generally. There's a real need for early diagnosis, as different types of dementia need different treatments. It remains a clinical diagnosis, and there's no single test for Alzheimer's disease. In fact, some individuals with elevated Alzheimer's disease biomarkers, such as Rihi mentioned, do not go on to develop mild cognitive impairment or dementia. It's also a growing problem as baby boomers age. As Rihi mentioned, age is a risk factor, and there's estimates, prevalence estimates as high as 33% of adults over 85 may have Alzheimer's disease. As he also mentioned, brain injury is another important risk factor. About 80% of traumatic brain injuries are what they call mild TBIs. They're also known as, often referred to as concussions. Mild TBIs are associated with typically about a twofold increase in the risk of dementia. However, there was a recent study, very large study with former professional soccer players that actually found more than a fivefold increase in Alzheimer's disease listed as a cause of death. I have often used eye tracking in my human studies, looking at trying to help people with clinical diagnosis, the progression of a disease, or evaluation of interventions. And I'm going to talk very briefly about each of those. I use an eye tracker, an infrared-based eye tracker, which gives a image of the eye and is considered state of the art. It gives very accurate spatial and temporal resolution down to milliseconds. And basically it can track people's gaze in real time. I use very simple tasks and measures. One task that I often use is called the prosecod. And I don't know if you're going to be able to see my mouse here, but basically the subjects look in the center here, and then a target appears, in this case, in one of 4 locations down here at the bottom. And the subject's task is just simply to make an eye movement as quickly as possible to the target, which is indicated by this little white arrow. And there's a lot of different measures. You can look at the latency, what's called the latency, which is the time between the onset of the visual stimulus and the initiation of that movement. And typically in a task like this, people are making these eye movements in 200 milliseconds, very fast. And so you can run 50 trials, collect data on 50 trials in a minute or 2. A second task that I often use is called the anti-saccade. You can run the exact same trials, but now you tell the subject, instead of looking to the target, to look opposite. So the correct response eye movement in this case would have been an eye movement to the top box. And again, you can measure the latency between the onset of the visual and the actual initiation of the movement. And in this case, the movements are more like closer to 400 milliseconds, so almost double in time. You can look at other variables as well. For instance, how many times they make an error and look to the target. That would be anti-saccade error rates. You could look at the amplitude of the movement. Does the eye movement fall short? The gain of the eye movement, the variability of the landing position, etc. There's a lot of different variables. I'm going to, this first task is considered a task that measures sensory motor function in the brain. Whereas the second task is considered a measure of cognitive processes, the primary one being, say, cognitive inhibition. So that's your ability to withhold or stop that automatic or reflexive eye movement to the target. But it also is a test of voluntary motor control, your ability to generate a voluntary movement to a place in space where nothing has actually appeared. It also includes working memory, so the ability to hold online the instructions to look opposite. And so I have used these tasks over the years, in some cases to help in diagnosis. So I'm going to give you an example. Parkinson's disease and multiple symptom atrophy are 2 Parkinsonian disorders that are very difficult for a clinician, if not impossible, to actually differentiate early on, and yet they require different treatments. So it's really important to differentiate them. And so we looked at these Parkinsonian patients, and the first thing I'm showing you here is that anti-saccade error rate, so that their ability, the number of times they make an error and look to the target. And you can see that both Parkinson's disease and multiple symptom atrophy patients are making many more errors on the anti-saccade test than age-matched healthy controls. So it's able to identify the Parkinsonian patients, oopsie. But if you look at prosaccade latency, then you see that you can differentiate between the two different Parkinson's disease and multiple system atrophy. So the MSA patients have longer prosaccade latencies, indicating additional degeneration earlier in the disease course. A second area that we've looked at is to try to examine progression. I'm going to again show you some data from Parkinson's and MSA patients. So we took these patients and we tested them not only at time point 1, but in a relatively short interval, 6 months, between first testing and a second testing, to see if we could see any difference in the disease progression. It is known that MSA has a much faster progression than Parkinson's disease, and we wanted to see if we could see anything. I'm showing you data from just the anti-saccade task, and I'm showing you both latency, that's the time to make that eye movement, as well as error rate. And I'm showing you here differences. So I'm taking time 1 and time 2 and subtracting it. So basically any bars above 0 are where the subject is faster at the second time point, and bars that are below 0, they're slower or worse. And so you can see that both control and Parkinson's disease patients at the second time point are actually a little bit faster and making fewer errors, indicating some learning at this very short time interval and or practice effects. But you can see that MSA patients are not showing that. So they either show no effect or actually greater errors by the second time point. So even within the 6-month time period, we're able to see evidence of indicative of more rapid progression of the disease in MSA patients. Finally, we've used eye tracking to look at evaluating interventions. So what I'm going to show you here is L-DOPA in Parkinson's disease patients. And I'm showing you, again, the anti-saccade error rates. And the black bar is Parkinson's disease patients in the off state, so more than 20, clinically defined, more than 12 hours without any L-DOPA. And then in the clinically defined on state after a dosage of L-DOPA. And what it's showing is that we can actually see that the Parkinson's patients with treatment with L-DOPA show a reduction in these anti-saccade errors, suggesting that in a medicated state, they're better able to plan and execute voluntary eye movements. Finally, we've taken these tasks to tablets, and I have a couple patents to try to figure out how to get millisecond timing on the tablets. And as a way of disclosure, I'm an owner of the company. And so these tasks, basically very similar tasks on a tablet, where the goal now is for the subject to actually move and point, touch the target or touch opposite the target. And using these more portable versions of the tasks, we've been able to identify mild TBI in the, whether somebody has a mild TBI or not, in the emergency room within 6 hours of injury. We've also been able to track more subtle cognitive changes following repetitive subconcussive blows in the field, such as like heading the ball in soccer. Finally, just a summary, these behavioral biomarkers are simple, quick, language independent. They're objective, sensitive, and reliable. They're difficult to cheat. They're clinically useful for diagnosis, including progression. So the goal would be to help differentiate Alzheimer's disease from these other dementias earlier in the disease course. They also would be possibly very useful in larger collaborative efforts to identify other laboratory or imaging biomarkers as a very sensitive behavioral measure and or evaluate interventions. I think the people following might have more information about grant funding, but this was one that actually Julia Chester had put up the previous year that I think would be relevant. Thank you.
Thank you, Anne. That's terrific. Really great overview of some powerful clinical tests that we could apply to human cohorts. So I think you really showed the rationale of these tests really nicely.
Thank you.
Okay, so then for the third presentation then from our representatives from Lewisburg. Beta, do you want to take do you want to be the one to start with the presentation?
Yes, can you hear me, Chris?
Yes, I can hear you perfectly.
Great, hello everyone. Apologies, we don't have slides. We wanted to keep this presentation a little bit more high level to give you an overview of Lewisburg Associates. I know that I've interacted with some folks on this call, but there are some of you which we haven't had the opportunity to connect with. So hopefully this overview of who Lewisburg is and the services we provide will be useful to you as you're thinking of strategy and engaging with federal agencies and potential funding opportunities. So just to take a few steps back about Lewisburg Associates, we are a full-service government relations and consulting firm. We're located in Washington, D.C. We have been interacting with Purdue University for about 4 years now. And what makes us different from, I guess, what you can say other folks in the community in DC is that we offer a wide range of expertise. So we have about 20 subject matter experts within our firm that represent various issue areas and federal agencies. I myself cover part of the HHS portfolio, and my colleague Nicole Parker, who will provide you with more specifics on some funding opportunities that might be of interest, is one of our NIH experts, and we thought it would make sense for Nicole to be on this call given the amount of funding behind Alzheimer's disease and other dementias and TBI as well. So just to give you a better sense of the type of strategies that we've rolled out in the past with Purdue, which I think would be of interest to this group, I like to classify the most effective strategies that we've pursued into 2 different buckets. Since we do focus exclusively on the federal level, a lot of our strategies are either tied to some sort of congressional ask or engagement with federal agencies. So this can range anywhere from tapping into the Indiana delegation in terms of creating a new program or funding opportunities, or asking for additional funding for a particular program that you've been able to take advantage of within NIH or other agencies. On the federal agency level, that's when we have come up with target lists, for example, of different federal representatives that you might want to meet with in parallel with the congressional strategy that we roll out. So in addition to building on the connections that you already have, a service that we provide is connecting you with program officers or other officials who you might want to engage with, especially before an opportunity comes out. So we're always listening in at advisory council meetings, and we're very involved in the communities and coalitions that have an agenda and push forward opportunities that you end up seeing coming out as a funding opportunity announcement on the NIH website, for example. So tying those together, I mean, this is an initial conversation. Obviously, we'd like to turn this into a somewhat iterative process, since this is the first time that we've coordinated or collaborated with this group. But one example off the top of my head, putting this more into context of those 2 different avenues that I just mentioned, is Lewisburg Associates, for example, has really tracked where the funding opportunities are. We know that Alzheimer's disease and related dementias, as well as aging policies and fundings behind those issue areas, that is a bipartisan topic, and we don't see that going away anytime soon. So we would play a role in identifying congressional champions within the Indiana delegation and beyond that we would want to share whatever creation or development, whether it be some new programming or support for existing programming that you receive to basically serve as champions to advance through appropriations or an authorization bill, further support, mostly more funding. And then in parallel, as I mentioned, we would want to socialize this with the federal agency, just because I know NIH especially doesn't necessarily like being told what to do by Congress. And I see Nicole smiling. I know that she's had experiences before. So obviously we would want to move a strategy forward in parallel to get buy-in from the right officials to make sure that we are successful with the strategy that we would want to roll forward. So that's a very preliminary initial idea. Obviously, we can turn this into another conversation, or hopefully a series of conversations where we can drill down into some sort of strategy that you all would be able to benefit from. So I'm actually going to turn it over to Nicole, who will provide you with some NIH funding opportunities that may be of interest. But let me add, I know that this is a team science initiative, and I mentioned that we have several experts that represent different agencies beyond HHS and NIH, including DOD, NSF, DOE, you name it, we have an expert. So we can always pull one of our specialists into a conversation as well to help you think about federal agencies that aren't necessarily traditional avenues that you've gone to for some sort of federal funding. That being said, I'm going to turn it over to Nicole, who will give you a snapshot of some NIH opportunities.
Thanks, Vita. So my name is Nicole Parker, and I help cover the NIH portfolio here at Lewisburg. And, like Vita said, we really do a great job of really tracking what NIH is doing, and other agencies, of course, what they're doing very closely. I'm not going to list specific funding opportunities for you all. I know you all are very familiar with grants.gov and can, find the opportunities that are available now. But what I really think it's important to share with you all is where we kind of think NIH is going. Vita shared earlier that Congress is very interested in Alzheimer's and related dementia research. Any disease you can think of that could touch someone very personally, like Alzheimer's or heart disease and things like that, will have champions in Congress. And so throughout the years, we've seen, especially with the recent BRAIN initiative that was passed earlier a few years back, we've seen just a consistent push for more Alzheimer's research. And that kind of lines up with the funding that these opportunities have. Chris mentioned earlier that the pay line for this type of research is much higher than the other opportunities, specifically at the National Institutes of Aging. So as far as the amount of funds for this type of research, there's definitely a lot available. I will say, at this particular point in time, NIH obviously is very consumed with COVID-19 research. A lot of people have asked, will that kind of limit the priorities in other areas? And what we know is that it will not. Even though COVID-19 research is taking a large priority, they're still very invested in these other disease areas that have plagued the community for many, many years. What I will say is that when you're thinking of how you're structuring your research, you want to think about how COVID-19 has specifically impacted Alzheimer's disease patients or other dementia patients, right? NIH is very interested in this particular community because, one, you all know that many patients that suffer from this disease area are located in long-term care services facilities where these outbreaks have taken place. So when we're thinking, if you're thinking about any human studies or things of that nature, you really want to focus on, well, how has COVID-19 kind of shifted what we're worried about in this sense? Because they're really going to be interested in looking into research related to that. Also, due to COVID-19, we've seen how it has impacted communities of color. Another thing that's very common in the Alzheimer's and related dementia research area is that I believe there's a lot of disparities on when diagnoses occur between certain populations. So when you're thinking of ways to diagnose Alzheimer's or other related dementias, thinking about how that differs between communities of color will be very important to NIH in the future. And I see more funding opportunities coming along in that area. In addition, related to that point, they're very interested in increasing diversity of clinical research participants in this area, and related to what you all have been talking about today as far as traumatic brain injury, understanding all the disease mechanisms that can lead to Alzheimer's disease and related dementias. That's something that's been recently mentioned as an area of interest for them all. One last point, because I know I don't want to go too over in time because you all have your breakout groups, but one last point that I really want to bring up is that in this current administration, in the Trump administration, there's been a huge focus on industries of the future. And so that's kind of pushed the interest in data science and artificial intelligence and all computational methods. We are starting to see at NIH a lot more research opportunities trying to bridge the gap. How can we use computational research to understand disease mechanisms or to diagnose patients or things of that nature? How can we use AI? How can we use electronic health records to kind of help, influence and, provide some outcomes and research outcomes? So things are moving, and I would say, and this is me kind of assuming, I think NIH hasn't really fully developed how they want to incorporate data science and AI into their research, but they realize it's something that they have to do. So one thing we started to see some opportunities come out recently, kind of connecting those two things, connecting data science, artificial intelligence to biomedical research. But we've also seen a lot of requests for information, kind of asking the community, how can we do this? So, you all, I think I read in the breakout groups, there's a computational group as well. So I think you all have all of the pieces. And so, think about how, not only what opportunities you can go after at NIH, but how can you kind of help frame the opportunities that will come out of NIH? There may be a lot of opportunities for you all to weigh in with program directors and program officers and respond to RFIs and kind of influence the direction of the research to give yourselves more opportunities. So I'll stop there. As Vita said, this is an initial conversation. We're available for further questions later on via email. And I'll also be popping into some of the breakout groups just to listen in and to see if there's anything I can add to the conversation.
Wonderful. Thank you both. I think I'm really excited to hear what you've just described. I'm happy to hear that we're already thinking along some of these lines, as you mentioned, an example being the computational breakout session, discussions earlier today also around Purdue's data science initiative. But I think we could really rely on your help in terms of how to properly prioritize and then really make the case in some of these areas or choose which ones we want to focus on in making that case. Are there any questions among any of the faculty attending here? We have some time to ask questions of Vita or Nicole if you'd like. I guess one question I would have is then, how should we continue? Should we organize ourselves up to a certain point and then engage with you sort of having organized at the institute level, or could anyone just email you and ask you questions? It suggests the latter would actually be appropriate. Is that correct?
Yeah, so something that I didn't mention is that we don't have a cap. So there's no limit on the services. You can email Nicole or myself. Chris, it sounds like this group regularly meets. If that is the case, we're happy to hop on to future calls if it makes more sense to have the whole group together rather than having one-off conversations. But we're accessible by email. And now that video conferences and Zoom has become a thing, I feel like we can do more face-to-face interactions as well. So feel free to take advantage of that as much as you need it.
Wonderful. I guess I'm also wondering, do work with faculty to help in responding to some of these requests for information? I mean, I'll admit, when I see requests for information, I'm usually scanning down to see what are the active programs, and there's a bit of an activation energy barrier, at least for me, to actually respond to an RFI. So do you provide some help for those kinds of tasks?
I can respond. Yeah, so it depends, right? So some RFIs, we can let you know if it's worth it or not, right? I do feel like there are some RFIs that for just only a university to respond wouldn't be worth it. It's more like a society should respond or something of that nature. But I would say if it's a research area that you're extremely passionate about, like for the reason why I mentioned that is because recently NIAMS, the probably inflammatory disease, arthritis, I always mess up their acronym, but they recently put out an RFI that said just that, like, how can AI influence our research portfolio, right? And so if you want to push the research agenda of your computational group in NIA, or it was to put out something like that, right? It would be worth responding to, and we can kind of help you figure out what you should be mentioning within the RFI. I think, as I'm a former scientist as well, and we can overwrite. We can write a lot of things that don't matter, but I think in RFIs it's really important just to get to the point. Even if they don't give you a word limit, I say 500 words is always enough. So yeah, we're able to help, but we'll also help you determine whether it's worth responding to as well, because all of them are not worth it. I would agree.
Yeah, that sounds really valuable. That's exactly what I was thinking. That I assumed you probably could give us some pointers on how to do the response effectively, because there's got to be some sweet spot there. Excellent. Any final questions?
Chris, I have a question. So
go ahead.
Nicole, you particularly can help people here at Purdue for NIH funding, but if DoD, would you be yourself or maybe another person would help?
Yeah, so for DoD, we would definitely bring in another person. I am not that familiar with the DoD opportunities, but we have several experts at our firm that work with DoD very closely.
Okay. Okay, yeah, that's good. Maybe at some point, Chris can collect these names. If it clearly relate to DoD, perhaps we can go to that person directly seeking help. Yeah. Thank you.
Ree, I think it's Laura. We've had her come speak at some of our meetings before, and you've been introduced, I believe, in the past anyway.
Yes. Yeah, I didn't see her name here, but I think this is for maybe for a group. If they're interested in DoD, then perhaps they can go to Laura. Thanks, Kira.
Okay, well, I'd like to thank Vida and Nicole for joining us and taking the time. This is really very helpful, and I also appreciate the suggestion that this could be the start of a conversation. We'll get organized to make sure that as we pursue this with you, that we're efficient with your time as well, and set up some initial priorities. Also, I'd like to thank Ree and Anne for great talks, really fantastic. I think that really gives us a lot of food for thought. And so now I'd like to invite everyone to go to whichever breakout session that you'd like to attend. The web links are in your instructions document, or they're also pasted, I think twice actually, in the chat box. And if you would like to start up a new breakout session that we didn't think of, please feel free to go ahead and start it on your own if you'd like, and press record, or ask me or Kata or Cynthia for help with that, and we can get you started. Thanks again for joining, and I hope that there are good discussions that follow. And we're a little bit late, so I think we can all spill over past 5 o'clock if you'd like. There won't be any hard stops in terms of the meeting rooms. And we'll post the information, and then we'll see you again at the end of the month for the next workshop, which is in the area of genetics and epigenetics more broadly. Thanks everyone for participating.
Great, thank you. I'm going to my breakout sessions. Thank you.
Thank you.